Hi, everybody, and welcome to our module on the cell cycle. Shown on the screen is the cell cycle. You can find this kind of drawing in most cell biology textbooks, and it shows the phases that a cell goes through as it grows and then divides to become two daughter cells. So if we start at this point right here, this is right after a cell has divided. At this point, cells enter what is known as the G1 phase, and the letter G is for growth. This is the first growth phase, and hence it is called G1. After G1, cells will enter the S phase. This is where they begin synthesizing DNA. There's then a shorter growth phase called the G2 phase, and this is followed by the M phase, which is when mitosis occurs and the cell divides into two daughter cells. In addition, some cells will exit the G1 phase and enter a quiescent phase known as G0. They will then sometimes go back into the G1 phase and then proceed through the rest of the phases. And then finally, I've cleared away my ink so I can show you that sometimes the cell cycle is divided into just two phases. The M phase is mitosis when the cell divides, and this entire phase where the cell is not dividing is called interphase. That includes all the time that the cell spends in G1, G0, S, or G2. On this slide, I've summarized the key points about the cell cycle. Interphase is when growth can occur. During the G1 phase, there is growth followed by the S phase where DNA synthesis occurs, followed by the second growth phase, G2. Cells can exit the G1 phase to enter the G0 resting phase, and then they can exit the G0 phase and go back to G1. And then the M phase is when mitosis occurs and the cell divides into two daughter cells. Now I'll go through each of the phases of the cell cycle in a little bit more detail. In the G1 phase, synthesis of proteins and cellular organelles occurs. The length of this phase is variable depending on conditions. If cells want to grow rapidly, this can be a very short phase. In other situations, this can be a longer phase. Mitogens are extracellular signaling molecules that are usually proteins that stimulate cell division. They function via molecules called cyclin-dependent kinases, which we'll talk about in a minute. And just some vocabulary here, a growth factor is a factor that stimulates growth in size. A mitogen is a factor that stimulates growth via cell division by driving cells through the cell cycle. Now, some molecules are both mitogens and growth factors, and the terms are often used interchangeably, but the technical definition is that mitogens drive cell division and growth factors drive growth in cell size. As I mentioned before, in the S phase, there is synthesis of DNA. Chromosomes will become a chromosome with two sister chromatids. And in the G2 phase, there is a short growth phase and preparation for mitosis. Now let's talk about the G0 or resting phase. This phase often occurs when there is an absence of mitogen stimulation of cells. It is a specialized non-dividing state, and it turns out that most cells in our body are in G0. Most cells in our body are not in the process of dividing. In fact, some are in permanent G0 state, others go in and out. For example, neurons and skeletal muscle cells are in a permanent resting G0 state. They are said to be terminally differentiated. They do not divide into daughter cells. Liver cells are usually in a G0 state, but they can divide if stimulated. You may know that the liver is a very regenerative organ, and if there is damage, liver cells can begin to divide and regrow the liver. Fibroblasts and lymphocytes can enter and exit G0 many times in their lifespan. They're often in a resting state, but when they are stimulated, they can begin to divide. And then cells in the bone marrow and GI epithelial cells and hair follicle cells are considered labile cells. The word labile means rapidly changing. These cells are rapidly dividing, and therefore they rarely or never enter the G0 phase. And for this reason, these are the cell types that are most commonly affected by chemotherapy. In the hematology section in the videos on chemotherapy drugs, I talk about these drugs which target rapidly dividing cells. This is helpful because they kill cancer cells, but these drugs also have adverse effects on these types of cells because they are rapidly dividing and are rarely in the G0 phase. And that's why side effects of many chemotherapy agents include bone marrow suppression, GI upset, and loss of hair. Now let's talk about mitosis. This is the shortest or most rapid portion of the cell cycle. It's usually divided into 5 phases called prophase, prometaphase, metaphase, anaphase, and telophase, and we'll go through all of these in the next few slides. The chromosomes in the nucleus of cells is usually relatively spread out, and this is helpful so that DNA can be transcribed. However, when cells enter mitosis, the first phase they enter is prophase, and in this phase the chromosomes begin to condense and pull together. In addition, an important structure called the mitotic spindle begins to form. This structure is made up of fibers of microtubules that will eventually pull the chromosomes apart into two daughter cells. In prometaphase, the chromosomes begin to organize on the mitotic spindle. This is a picture of prometaphase shown on the screen here. And then the hallmark of metaphase is that the chromosomes line up in the middle of the cell, as shown in this picture here. They are lined up on what's known as the metaphase plate. From here, they are going to eventually be pulled apart to opposite ends of the cell. And then in anaphase, the chromosomes separate to opposite ends of the cell. Remember, in the S phase, the chromosomes are replicated into two sister chromatids. Those chromatids are now pulled to opposite ends of the cell in anaphase. And then finally, in telophase, the spindle breaks down and the cell begins to divide. That's called cytokinesis. And then you are left with two daughter cells that came originally from one single cell. Everything I've talked about up until now is pretty basic. You may have had it in a college biology course. But what you might be less familiar with, and what it's very important you understand for the USMLE Step 1 exam, is how cells control progression through the cell cycle. Cells in our body regulate progression through the cell cycle by regulating progression through checkpoints. These are important parts of the cell cycle, also called restriction points, that the cell does not move beyond unless conditions are just right. So for example, the G1 to S phase transition just prior to S phase entry is a very important checkpoint. So is the G2 to M phase transition just prior to mitosis. In addition, there is an important checkpoint within the M phase itself prior to anaphase and cytokinesis. And what the cell does is look at various regulatory elements and it arrests itself if conditions are not appropriate. In other words, it does not proceed further in the cell cycle if conditions are not just right. And in particular, the first checkpoint in late G1 at the G1 to S phase transition is very important because this is where the cell commits to proceeding into the cell cycle and growing. In other words, once the cell moves beyond this checkpoint, it is entering the S phase and beginning to synthesize new DNA, and therefore it is committing to dividing into two daughter cells. So the first thing to be aware of about all of these checkpoints is that they depend on cyclin-dependent kinases, which I mentioned before. These are the central components of cell cycle control. They are kinase enzymes, which means that they lead to phosphorylation of other proteins. Any kinase enzyme phosphorylates something else. And they are always present in the cell, but they are inactive. So in other words, they are waiting to drive the cell through the cell cycle, but they're inactive, and they need other molecules called cyclins in order to become active. Therefore, this means that cyclins are regulatory proteins that activate the cyclin-dependent kinases. And cyclin CDK complexes phosphorylate regulatory proteins, and they allow cells to progress through the phases of the cell cycle. There are many different classes and subtypes of cyclins that have been described, and all of their levels vary during the cell cycle. Shown on the screen is a picture you can find in many cell biology textbooks. This is the G1 phase on the left. Here's S phase, G2, and mitosis. And you can see that levels of the various cyclins are going up and down. And when the level goes up, that cyclin will activate a cyclin-dependent kinase, and that will drive the cell through the next phase of the cell cycle. So now let's focus on the very important G1 to S phase checkpoint. Remember, this is when the cell commits to DNA synthesis and cell division. So during the G1 phase, cyclin-dependent kinase activity is suppressed, but mitogens can activate cyclin-dependent kinases and drive entry into the S phase. The way they do this is by interacting with cell surface receptors. This will activate intracellular pathways and increase G1 cyclin levels, and those G1 cyclins will increase cyclin-dependent kinase activity. In the G1 to S phase checkpoint, cyclin CDK complexes activate proteins called E2F proteins. These are transcription factors that bind to DNA promoter regions and activate genes that are necessary for entry into the S phase. Now, E2F proteins are normally inhibited, and the reason they are inhibited is because they are bound to molecules known as retinoblastoma proteins. This inhibition is released by the G1 to S phase CDK phosphorylation of retinoblastoma proteins. I'll show you a picture of that in a minute. What this means is that retinoblastoma proteins regulate cell growth, and therefore they are called tumor suppressor proteins. So here's a picture to make this very easy for you to understand. Normally, the E2F proteins are inactive, and the reason they are inactive is because retinoblastoma proteins, which are active, are bound to the E2F transcription factors. When cyclin CDK complexes come in, they can phosphorylate the retinoblastoma proteins. This will release the retinoblastoma proteins from E2F, and then the E2F transcription factors can go and drive cell growth. So the retinoblastoma proteins and the E2F transcription factors are one element of the G1 to S checkpoint, but there is another important element of this checkpoint, and that has to do with DNA damage. If DNA damage occurs in the G1 phase, this can arrest cell division and prevent progression into the S phase. This allows time for repair and it prevents the development of mutant cells or cancer cells that have abnormal genes. What this means is that DNA damage has its own ability to initiate signaling pathways that affect whether or not a cell proceeds beyond the G1 to S checkpoint. There are two pathways by which a cell can become arrested at the G1 to S phase checkpoint if DNA damage has occurred. The first one is called the ATM pathway. This pathway is activated by double-stranded breaks in DNA. ATM stands for ataxia-telangiectasia mutated, and it gets this name because the ATM gene is mutated in the disorder ataxia-telangiectasia, which I discuss in the neurology section in the video on the cerebellum. The other pathway is called the ATR pathway. This pathway is activated by single-stranded breaks in DNA. Both pathways lead to phosphorylation of proteins, and via phosphorylation of proteins, both pathways can cause cell cycle and growth arrest if there is DNA damage in the G1 phase. The p53 protein is the major target of the ATM and ATR systems. It becomes phosphorylated when there is DNA damage, and this prevents breakdown of the p53 protein and increases its levels and activity. Basically, the p53 protein easily falls apart unless there is DNA damage, and then it becomes stabilized. When p53 is stabilized, it induces transcription of a protein called p21. p21 can bind to cyclin-dependent kinases and inhibit their activity. And remember, it is these kinases that allow progression through the cell cycle. So when they are inhibited, this will block cell progression through the cell cycle. What this means is that both p53 and p21 are considered tumor suppressors because they prevent abnormal cell growth. So once again, I'll use a picture to make this very clear. When there is DNA damage, the p53 protein, which is normally unstable and rapidly breaks down, becomes stabilized. It can then go and promote transcription of the p21 gene, which will lead to synthesis of the p21 protein. And the p21 protein can inhibit cyclin-dependent kinases and lead to growth arrest. And just to tie this into what we talked about before, earlier I showed you how cyclin CDK complexes can release the inhibition of E2F so that E2F can go and promote cell growth. What p21 does is block this process, and it prevents cell growth, and therefore the cell does not proceed through the G1 to S phase checkpoint, and therefore does not continue growing and dividing. And here I've made a summary slide just to make this very clear. So the G1S checkpoint is very important because that's where the cell commits to DNA synthesis and cell division. If there is mitogen stimulation via the retinoblastoma E2F pathway, that can drive the cell from the G1 into the S phase past the G1S checkpoint. On the other hand, if there is DNA damage at any point during the G1 phase via the p53 pathway, this can arrest the cell in the G1 phase. I'll finish this video by going through some of the classic clinical correlations of the cell cycle. These are all very high yield to know for your board exams. We'll start by talking about retinoblastoma. So retinoblastoma is a rare childhood eye malignancy, and it's caused by mutations in the RB1 gene. This gene codes for the RB protein, and if children have an abnormal retinoblastoma protein, then they will have unregulated cell growth. So just like we've been talking about, the job of retinoblastoma is to hold back E2F. So if you don't have retinoblastoma, then E2F will lead to uncontrolled cell growth. There are rare inherited forms of retinoblastoma, and in this situation, the child is born with one mutated RB gene in all of the cells in their body. So initially, the child is heterozygous with one mutated gene but one normal gene, and so the child won't have cancer. But then if something comes along and takes out that normal RB gene, the child loses heterozygosity, and this leads to cancer. And so this term, loss of heterozygosity, is used in cancer biology and cancer research to describe when a cell goes from having one abnormal and one normal copy of a gene to having two abnormal copies, and that leads to cancer. The Li-Fraumeni syndrome is a rare syndrome where patients develop multiple malignancies at an early age. It's sometimes called the SBLA cancer syndrome because some of the malignancies that can occur include sarcoma, breast cancer, leukemia, and cancers of the adrenal gland. The cause of Li-Fraumeni syndrome is a mutation in the tumor suppressor gene TP53, which codes for the p53 protein like we've been discussing. So when there's a mutation in this tumor suppressor gene, then the cell cycle is not arrested to allow for DNA repair because p53 function is abnormal. As a result, cells of patients with the Li-Fraumeni syndrome fail to undergo apoptosis as they should, when there is DNA damage. And so over time, they accumulate DNA damage, and that leads to malignancies in all these different organ systems. The human papillomavirus is a sexually transmitted infection that's associated with cervical cancer, and it expresses an oncogene called the HPV oncogene E6, and this leads to synthesis of an E6 protein. Well, what E6 does is it binds to P53, and this leads to P53 degradation. So once again, once P53 is lost, cells can undergo uncontrolled growth, and that's what leads to cervical cancer. The way the P53 protein is degraded in cells infected with the human papillomavirus is through a cellular process called ubiquitination. It's high yield to know this for your boards. In this process, P53 becomes bound to the cellular protein ubiquitin, and this targets the P53 protein for degradation in proteasomes. And then finally, in the endocrinology section, I talk about a rare form of thyroid cancer called anaplastic thyroid cancer. And you should know that cancer cells in this form of thyroid cancer are strongly associated with mutations of the P53 gene. These cells often produce inactive P53 protein, and as we've been discussing, if you don't have P53 working, that leads to uncontrolled cell growth. And that concludes our module on the cell cycle.